everyone, welcome to my channel Pure Biology, and today I come with another video that is the targeting of mitochondrial proteins into mitochondria. And this is obviously before this I have already explained the structure of the mitochondria in my previous video, so check out that one. So let's continue. Here the how the targeting of the matrix protein is possible in mitochondria. So most of the matrix targeting protein that enter into the mitochondrial matrix contain N-terminal cleavable matrix targeting sequences. Okay, so maximum proteins that present in the mitochondria or generate transfer or translocation of the mitochondrial protein. What kind of mitochondrial protein is translocated? You can say that are N-terminal cleavable matrix targeting sequences. These are called the pre-sequences, and here the two complex very important. One is the TOM complex, another is the TIM complex. So I have already explained these two complexes in my previous video, so check out. Here I just explained the process, what is the actual processing occurring here. So here main these are synthesized. What are synthesized? The matrix targeting proteins. These are synthesized by the ribosome, because you all know that ribosomes help in the protein synthesis. So specifically 80S or 80 subunit ribosomes, these are very important for the synthesis or for the synthesis of this polypeptide chain or this kind of proteins. And these are actually remain in the mitochondria in the form of unfolded manner or loosely folded manner, as they can pass through any complex. And here this carried out by the HSC, that means here the role of the HSC70 is very important. Okay, HSC70 play important role in this processing. Next one, first what is happening here, this TOM complex or during translocation, this MTP means matrix targeting protein, this bind with the TOM complex, and TOM complex is one kind of TOM20 generally is the main TOM complex that can recognize the particular protein or polypeptide chain, or you can say the pre-sequences. Okay, and then these transfers these pre-sequences or precursor proteins where to the receptor, or you can say the TOM22. Okay, and this mitochondrial sequence, this direct the proteins, that means this after entering into the TOM22, it can try to enter TIM23 complex. Okay, and here TIM23 complex that is associated with the pre-sequences, that means that containing the TIM23 with which the pre-sequences already bind that contains translocase associated motor. Okay, translocase associated, that means TIM23 has pre-sequences translocase associated motor. Okay, so here this motor is another complex or another factor, you can say, because motor contains consists of the different proteins that are MTHSP70 and CO chaperones and nucleotide exchange factor and other proteins. And here this mitochondrial HSP70 that binds to the translocating polypeptide chain. Okay, that generally bind to the polypeptide chain and drives its movement to the matrix. So what is the role of HSP70? That is present within the motor, or you can say the pre-sequences translocase associated motor. So this motor containing this HSP70, it has important role because it binds to the translocated polypeptide chain and drives its movement to the matrix. Okay, and here the hydrolysis of ATP is very important role, because this matrix interaction cycle requires the hydrolysis of ATP. Okay. So where the pre-sequences just after entering or soon after the pre-sequences that enter into the matrix, it is removed by the mitochondrial processing peptidase. Okay, this is another enzyme, peptidase. So this peptidase that remove the pre-sequences just after entering into the matrix, and empty HSP70 and many matrix proteins that are passed on to another chaperone protein. This is called the empty HSP60. That means up to this, the role of empty HSP70, this is seen, but after that the empty mitochondrial HSP60 that plays important role. Okay. So what is the role of this? Its main role is that it helps the unfolded polypeptide chain to fold. That means from here to this portion, the polypeptide chain that remains in unfolding manner, but when it enters into the matrix, it is it generally transform into folding manner with the help of HSP60 or mitochondrial HSP60. Okay. So this process, the translocation of the proteins, this is an active process, and in addition to ATP hydrolysis, an electrochemical gradient across inner membrane is required. So here it is also said that the hydrolysis of ATP is very important or required here. So it is again mentioned here that hydrolysis of ATP is necessary here. Okay. So this is the topic that targeting to the mitochondrial proteins into the mitochondria, and TOM complex and TIM complex have important role. Okay. Okay. So here the targeting to inner membrane is explained now. So here what is happening? The targeting of proteins to the inner mitochondrial membrane that requires the different strategies. Okay. Strategies. Okay. Strategies. So in most single pass, generally two kinds of membrane proteins. One is single pass, another is the multiporous. Okay. So here the single pass transmembrane proteins or the N-terminal cleavable matrix targeting sequences first direct the precursor proteins into the matrix. First point it is that directs the precursor proteins into the matrix. Clear? And these precursor proteins that are actually this contain a stop transfer signal. Okay, this is this precursor proteins that is directed into the matrix that contains a stop transfer signal. Okay, this stop transfer signal is located just after the mitochondrial pre-sequence. Okay, just after the mitochondrial pre-sequence. And what is the role of this signal? This signal actually arrests the translocation of the mitochondrial inner membrane protein. Which protein? The precursor proteins that being released literally in the lipid layer. Okay. So what I am saying here, that the precursor protein, generally this precursor protein that enter into the matrix, this contain one kind of signal. This signal is actually, what is the function? Arrest the translocation of mitochondrial inner membrane, arrest the translocation in the mitochondrial inner membrane with the precursor proteins being released literally into the lipid layer. That means that precursor proteins which are released into the lipid layer that are not translocated, these signal arrest the translocation of this type of precursor proteins which are being released into the lipid layer. Okay. First, N-terminal. How is it possible? The N-terminal cleavable matrix. Sorry. Okay. So first, N-terminal cleavable matrix targeting sequences that is recognized by TOM20. That is the one substance you can say, or component you can say of the TOM complex. Okay. Then this protein is translocated through the TIM23 complex. Another protein translocase, OXA complex, it is also held in mediates, you can say, the insertion of those inner membrane proteins that are synthesized within mitochondria by 70S ribosomes. Okay. So what is saying here, that the actually protein sequences that are recognized by the TOM complex, that is the TOM20, and this protein is translocated in through this TOM23 complex. First TOM20, then from TOM20 it is transported towards the TIM23, and another translocase or another enzyme you can say translocase, OXA complex, it also helps in the translocation of proteins. Which proteins? That are synthesized by the 70S ribosomes. Okay. Now here to insert some important inner membrane proteins that are initially transported into the matrix space by other complexes. So here it also helps, it means here this OXA complex, okay, also it helps to insert some important inner membrane protein that are initially transported into the matrix space by the other complexes. So if any protein that is already transported into the matrix space, this is also translocated by the OXA complex. Okay. And in case of multipore proteins that contain internal targeting sequences, and these are totally recognized by the TOM70, and after recognition this TOM70 transfer this to the TIM22, and here the TIM22 that includes different small TIM substances or TIM subunits, 8, TIM8, TIM9, TIM10 and TIM13. So these also help in this translocation. So what is saying here, you can totally understand that first this terminal cleavable matrix targeting sequence is recognized by the TOM20 and it is translocated through the TIM23. Some cases OXA complex has an important role, or this plays an important role for this translocation, that are generally synthesized by either 70S ribosomes or other complexes and that present in the matrix spaces. Okay. And in case of this is possible only in case of what kind of proteins? These are the single porous or single pass protein. But in case of multiporous protein, that are generally recognized by the TOM70 and this translocated through the TIM22, and this TIM22 that containing TIM8, 9, 10 and 13, these play important role for this translocation. In case of outer membrane, which are playing the important role, obviously TOM complex play important role, because without TOM complex the recognition of the sequence is not possible, and after that the TIM complex either 9 or 10 or 8 or 13 play important role in the translocation. Okay. And also another protein or another complex, this is the SAM. Okay. SAM, that means the sorting assembly machinery complex. This also help in this translocation of the outer membrane protein, okay, or outer membrane pre-sequences. So these are the actually the transportation or the translocation of membrane protein, that is outer membrane protein, inner membrane protein, or matrix proteins. Okay. So I will explain another topic in my next video. So continue with me.